be in position to see your screen?
Yes, sir.
All right.
Yes.
Okay. So yesterday we got an interruption and we never covered fully what we had to cover. So okay. I was saying that we are going to continue from where we stopped yesterday. I remember that yesterday we talked about, we described the cell membrane, but I don't exactly remember where we stopped because I remember I went off. And there's somewhere where I had stopped, but I don't recall. Anyone to tell me?
We had started on the endoplasmic reticulum.
Okay, all right. Okay, so the endoplasmic reticulum is a very important organelle in your cells. And this endoplasmic reticulum, it's, I said it's categorized into two. It's categorized into the rough endoplasmic reticulum and the smooth endoplasmic reticulum. These names are given such way because the rough ER has a granular appearance because of these structures that look like granules around it. Though these are not granules, these are just ribosomes that are on its outer surface. And then this lacks those ribosomes. That's why it's called smooth endoplasmic reticulum. And then I said this rough endoplasmic reticulum, since it has that communication with the ribosomes, it's going to also contribute towards protein synthesis, and then afterwards it's going to allow transportation of the proteins through its flattened structures, as you can see them here. It will transport those proteins eventually up to the Golgi apparatus. Okay. So that's the function of the rough endoplasmic reticulum. Then the smooth endoplasmic reticulum, I said, it's going to contain abundant enzymes that are involved in lipid synthesis. And I explained to you that in your body, lipids are needed. Yes, we can get those lipid components from the food that we eat, but also your body itself is structured in a way that it has components in the cells that can produce those lipids. Okay. So among the lipids, I said we have cholesterol, phospholipids, okay, and these components are all going to be part of the cell membrane, as we discussed last time. Then some people are not muting. I don't know why. Kindly, let's mute our devices. Let's all mute our devices. Okay. So I was saying that that smooth endoplasmic reticulum will help in the production of lipid components in your cells like cholesterol and phospholipids. And I explained that cholesterol is very important because it's going to be used to build cell membranes. Phospholipids also builds, it's part of cell membranes. Additionally, cholesterol is going to be used in your body mostly in the cells in the adrenal cortex to help in production of sex hormones. Okay, and additionally, the phospholipids are going to further be utilized, for example, in the cells of the liver to help in formation of bile salts. Okay, phospholipids and also cholesterol, they are going to additionally all going to be utilized in the cells of your liver to help in formation of bile salts. Bile salts are very important in emulsifying fats so that the fats can be utilized in your digestive system. Okay, without the bile salts, the fats cannot easily be digested. So those bile salts, for them to function normally, they have to be made up of phospholipids and cholesterol. And those components, this is where they come from the smooth endoplasmic reticulum. Then also I said this smooth ER can also contribute towards detoxification in your cells. But there's a main organ of detoxification as we are, there's a main organelle of detoxification as we are going to see shortly. Okay, so I said all those produced products are going to be sent to the rough endoplasm, to the Golgi apparatus so that it can package them. And after packaging them, what is going to happen is that they are going to be sent out of the cells to different parts of the body. And again, I'm going to use an example of insulin because you all know it. Insulin is a protein produced in the beta cells in the pancreas. Okay, so after the cells producing it, the Golgi apparatus in the beta cells of the pancreas will package that insulin and release it so that it can be deposited in blood to be transported to the different organs like the liver to help in blood sugar regulation. Okay, so that's what happens at the cellular level. So all these organs, all these organelles are very, 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 very critical and very important. Okay, so lysosomes. Lysosomes are organelles that are going to be also very important in your cells. These lysosomes are going to be scattered all over the cytoplasm, as you can see them here. They are scattered all over the cytoplasm. Here we are seeing one is labeled. You can see another one there. So there are many all over the cytoplasm. And these lysosomes are going to be containing enzymes. These enzymes, we call them hydrolytic enzymes. These enzymes are called hydrolytic enzymes. And these lysosomes are going to mostly be abundant within white blood cells. White blood cells are very many in your body. Among the many, we have macrophages, neutrophils. All those cells have abundant lysosomes. So what happens, those cells, we all know that they carry out phagocytosis. And always after phagocytosis, a pathogen is going to be taken in. It can be a bacterium, it can be taken in, can be eaten by this big white blood cell, which can be macrophage. And after this pathogen entering, it's going to be destroyed with the help of the hydrolytic enzymes produced by the lysosomes. And that overall will help in preventing bacterial infections within your body. So this is very important. And this reminds me of the diseases that can occur in cases of deficiency of some hydrolytic enzymes in the lysosomes. Those diseases are called lysosomal storage diseases. I want you to read about them. Read about the different lysosomal storage diseases, their causes, and the presentation. in patients with those lysosomal storage diseases. They are not so that common, but you can find a patient with such a disease. And if you had known about it, it can be easy for you to easily diagnose this patient. You can easily tell the presence of that condition. So read about what we call lysosomal storage diseases. This slide is further showing you the mechanisms used by lysosomes within our cells. These lysosomes are, we said, made up of hydrolytic enzymes, and whenever a bacterium or any pathogen is taken in, let's say I show this is a macrophage here, this whole cell is a macrophage. Whenever a pathogen is taken in, hydrolytic enzymes are released onto that pathogen so that it can be destroyed. Okay, this mechanism is called heterophagy. It's called heterophagy because these lysosomes are feeding onto components that are coming outside, that are coming from the outside of the cell. That's why we call it heterophagy. Then there's also what we call autophagy, and in autophagy, what happens? Lysosomes help in recycling of worn-out organelles. It can be any organelle. In this example, we are seeing a mitochondrium. Mitochondrion may be past its lifespan, and there can be a need of recycling it so that more mitochondria can be produced in your cells. So what happens? Lysosomes are going to attach onto that old organelle and then release enzymes to allow recycling of that organelle. We call this autophagy. The lysosome is feeding on, or it's destroying components within the cell where it's also situated. So it's autophagy. Okay. Then there is another mechanism that we call autolysis. Autolysis. It is not displayed here, but basically autolysis, it's a mechanism that can be used. For example, let's assume you have viral particles in your body. If these viral particles enter your body cells, we all know that viral particles can utilize our cellular machinery to increase in number within our cells. So whenever those viral particles accumulate in the cells, lysosomes in that cell can help kill that cell so that those viral particles can also be destroyed to prevent them from spreading to other cells of your body. So we call that autolysis. Lysosomes can initiate destruction, complete destruction of a particular body cell where it also resides, as long as that cell is invaded by multiple viral particles. That is autolysis. Okay. So there are three mechanisms: autolysis, heterophagy, and autophagy. Those are all mechanisms of lysosomal function. Peroxisomes. Peroxisomes are also very important organelles in your cells. And these peroxisomes, they are also made up of enzymes, just like lysosomes, though here the enzymes are oxidative enzymes. These enzymes include peroxisome oxidase, we have peroxisome catalase. I'm not going to go into detail of how they work, but understand that these enzymes are going to help neutralize toxins or toxic metabolites within your body. Okay, and because of that, we can appreciate that these peroxisomes are more abundant in the liver cells. They are more abundant in the liver cells. And whenever you consume things like alcohol, you all know that alcohol is further metabolized in your body to form other metabolites. And those toxic metabolites from alcohol they can easily be neutralized by the peroxisomes within your hepatocytes. Okay, but an individual who consumes large amount of alcohol obviously may not easily be saved because the the number of peroxisomes in the liver in the liver cells may not be abundant enough to neutralize all the toxins that may be coming in from the too much alcohol that you're taking. And that's why over time, individuals who chronically take alcohol over time tend to get liver disease because the too much accumulated toxins in the liver tend to target and destroy the liver tissues, resulting into liver disease and many other complications on the liver. Of course, the worst is hepatocellular carcinoma or liver cancer. So those are the peroxisomes, and that's what they do within your mitochondria. Mitochondria, these are organelles found in your cells. You can see them here. These organelles are going to be very important in your cells. They are made up of finger-like projections or extensions. You can see the inner membrane has those finger-like extensions that we call a cristae. These extensions are made up of different proteins or enzymes that are going to be involved in breaking down different biomolecules, for example, glucose. Okay, the glucose that you obtain from the carbohydrate meal that you've taken, let's say you've had bread in the morning, the glucose that's going to be obtained after breakdown of that bread in your body, that glucose will enter your cells together with the oxygen that you're taking in. That glucose can be broken down with the help of the enzymes lying over the cristae, so that energy can be yielded in your cells. That energy is very important to allow cell division, for example. So you know that always your cells are continuously dividing. Of course, some cells, as we are later going to see, some cells are always continuously dividing, and this continuous division can't happen without ATP. Okay, so there are many ways how this energy is relevant within your cells. There are some components that may need to enter your cells, but they can't enter without ATP. Okay, so this ATP is very, very vital within your cells. And it's generated by this structure that we can call the powerhouse of the cell, structure that we call the mitochondria. So we've almost talked about most of these organelles. I think what we have not talked about, let me see, we've not talked about the cytoskeleton. Now, the cytoskeleton is just a network of protein structures which are going to be situated close to the cell membrane. You can see those network of protein structures, those strands there. Those are strands that make up the cytoskeleton. Of course, some are called microtubules, some are called intermediate filaments, some are called actin filaments. They all have different functions. Okay. For example, if we are to talk about the actin filaments, these actin filaments, they're going to be more abundant in cells, for example, the muscle cells. And those actin filaments are important
the audio is hard. Not getting any audio. Now let's say disconnection.
I think it is a disconnection from Sir. He might be having some challenges. Let's first wait.
The network is not all that good.
Excuse me, sir, you're muted to some of us.
Sir, we can't hear you.
Excuse me, sir. Good morning. We are not getting you, please.
Sir, you're muted. You're muted. Please unmute so that we can hear you, sir.
Maybe someone can give him a call.
Anyone listening, sir? Because you're not getting any information.
Guys, let's be a little patient. Maybe he's knowing what is going on.
Hello?
But we can see the cursor moving, which means he's trying to explain,
Can
but
you
he's
now
not
hear me?
aware.
Now we
Yes,
can
we can
hear you.
hear. We can hear you, sir. But unfortunately, I can't see
Hello?
the presentation. Hello, sir. I can hear you, but I'm not able to see the presentation.
Okay, so can you now hear me?
Yes,
Yes,
we
sir.
can hear
Yes.
you.
Yes, sir.
Okay, that's one of the challenges of online sessions, but I know we can overcome. So
But
come
but I
back.
still have a challenge, sir.
The screen is
Uh,
visible that side, isn't it?
Mine is not visible,
Yes.
sir.
It is there.
If it's visible.
Yes,
It's
it is
visible.
visible.
Okay.
Mine is not visible.
Refresh your page, please. It might actually be network.
It is network problem, I think.
Okay.
Today network
All
is
right.
too.
All right. Let's refresh. If you can't hear me, if you can't see the screen, you can just simply refresh. Me too. After the call, I couldn't hear you, and you couldn't hear me. I just refreshed. Within a few seconds, I was back. Okay. So I was mentioning that the cytoskeleton overall, it's a network of protein structures that are going to be supporting these organelles, but also maintaining the shape and the structure of your cells. Okay. Remember, your cells don't have the cell wall like the plant cells. Plant cells have the cell wall, and that's enough to support the structure of the cells. But in our cells, what helps in supporting the structure and the shape of the cells is the cytoskeleton. And then I was adding something saying that the cytoskeleton in some particular cells of the body further plays different roles. For example, I was saying that there are some cells of the body that have cilia. For example, we have the cells lining the epithelium of our respiratory tract has got cilia. The cells lining the epithelium of the fallopian tubes has got cilia, and those cilia are just finger-like extensions that are made up of microtubules. Okay, and because of the microtubules, those ciliary structures can easily beat sideways to allow probably propulsion of any substance that may be in that particular part of the body. Then also I was saying that sperm cells, we know that do have a tail. That tail is called a flagella, and that flagella is made up of microtubules, which is part of the cytoskeleton, and that gives the sperm cells the ability to easily swim through the vaginal fluids after ejaculation. Then I'm also saying that the actin filaments are also part of the cytoskeleton, and they are abundant within the muscle cells, or what we call muscle fibers, to allow easy muscle contraction. Okay, so that's just the summary of cell organelles. I've just summarized the organelles, but you can find a lot more details in the textbook that I showed you. And last time I told you, you always have to use textbooks, because we use textbooks when setting questions. These slides just provide a summary that can help me to mention what is important. So those are cell organelles, and now let's switch and look at cell cycle and cell division. Okay. Okay, there is a hand from someone. There's a hand from Rose. Misanya Rose. Misanya Rose, you can unmute and speak.
Good morning, sir. I would like to know that, yes, I would like to know the difference between many parts of a cell than the principal parts of a cell.
There's no difference. There's no difference. It's just the English that you're using. So if someone says main parts of cell, principal parts of the cell, all that means the same. Okay, that person is talking about the contents of the cell, the organelles of the cell, the functional parts of the cells, and those are the components that we've just talked about. Ms. Sanya Rose, hope that is clear. Nicholas, very fast. Nicholas.
Yeah, thank you very much. My concern is about we looked at peroxisms, and we looked at specifically oxidative enzymes that do neutralize the toxins. I would like to know the examples of those enzymes specifically. Thank you.
You'd like to know, I beg your pardon, examples of?
The enzymes that do neutralize the toxins, the oxidative enzymes.
So within the peroxisms, I mentioned that they are catalase enzymes. Okay, catalase enzymes that are going to help in detoxifying components like hydrogen peroxidase. Okay, within the cells. There are many other enzymes, for example, peroxisome oxidase enzymes, okay, and most of other enzymes, you learn more about them when you're studying biochemistry, okay. There's a course unit of biochemistry. I don't know, I think in second year, okay, but those are some of the peroxisome enzymes, okay. Is that clear to you? The catalase, the oxidase. The oxidases are many. We have acyl-CoA oxidase, hydroxyl acid oxidase, urate oxidase, xanthine oxidase. There are many. We can't mention all of them. Okay. They are named based on what they are going to break down or what they are going to neutralize. Is that clear? Who was that? Nicholas, is that clear? Okay. Michael, very fast, let's hear from you.
Yes, thank you, sir. I'm getting you clearly. I think my only challenge is still the presentation, which is not being showed on my side here. That is why I've been
The screen is
raising my
not.
hand thing.
The screen is not being shown that side.
Yes, sir.
After multiple times of refreshing, still it's not there.
No.
Okay, so now that means that maybe that area where you are now, like maybe that's where you're sitting now, the network is not good. And next time maybe you try switching positions. Probably the network strength is not good where you are now, Michael.
Okay, thank you, sir.
Mumbere Samuel.
Thank you, sir. My question is about the materials that pass through the plasma membranes. Those ones that are not supposed to pass through, because I read somewhere they said glucose is one of them, so I don't know whether it's true or not. So I would like to know the materials that don't pass through the plasma membrane.
Okay, so the plasma membrane, last time we said that it is largely made up of lipids. Lipids are non-polar molecules. That means they are not charged. Lipids don't have positive and negative charges on them. They are non-polar. Okay, so if there is any molecule that is non-polar, that molecule can easily pass through the cell membrane, as long as that molecule is small enough. Of course, larger ones can be prevented, but small non-polar molecules, for example, the respiratory gases, oxygen, carbon dioxide, they are non-polar. Oxygen is not charged, carbon dioxide is not charged, and they are small molecules. They can easily permeate through the cell membrane of the cells of the lungs during gaseous exchange. So that is one, small non-polar molecules. They can easily go through. Then two, lipid-soluble molecules. As long as a molecule is lipid-soluble, it will easily dissolve in the through that lipid core and is either enter or leave the cell via the cell membrane. And examples of these lipid-soluble molecules, yesterday I told you the fat-soluble vitamins, the vitamin A, vitamin D, vitamin E, vitamin K. Those are fat-soluble vitamins. They can easily enter and leave your what? Leave your cell. Then alcohol. Alcohol can also alcohol can also easily enter your cells, and probably that may explain the reason as to why whenever someone takes alcohol, alcohol easily enters every body cell, probably contributing to the symptoms associated with someone who is has taken large amounts of alcohol. So alcohol can also easily go through the cells via the cell membrane. Glucose is a large molecule. Glucose is a large molecule. Amino acids are large molecules, and those will utilize the transporters on the cell membrane, the carrier transporters or the carrier proteins. Okay. Ions, for example, sodium ions, calcium ions, what else? Potassium ions. Those ions, yes, they are minute components, but they may not easily go through the cell membrane because they are charged. Remember, the lipids are not charged. So if there is anything that is charged with either positive or minus, it can't easily go through the membrane. That's why those ions, the sodium, the calcium, all those ions, they will have to use their particular or specific ion channels. Okay, so those are the criteria that you can note, criteria that determine the molecule that will easily go through the cell membrane. But as I commented yesterday, this whole concept of membrane transport can be appreciated. There's a chapter in that textbook that can help you appreciate all these concepts. Okay, very well. I don't know if I have answered you. Was it somewhere? Mumbere somewhere?
Yes, you've answered. I've understood.
Okay. Okay, I think let's continue. I may not be able to address all the questions. I gave out my number so that if at all maybe you have anything that I have not attended to in the session, you can just text so that I can easily reply to you. Okay. Kindly allow me continue so that we can look at something regarding cell cycle and cell division. More questions about the cell structure and function, you can just simply text me. If you have any question, you can text and I can easily access my WhatsApp and reply to you. Okay, cell cycle and cell division. Now, you all know what cell division is. As you hear the word division of cells, process by which a cell divides to give rise to daughter cells. That is cell what? Cell division. And this process is very, very important in your body. Without cell division, it will be hard for you to grow to the size that you have now. For you to grow from infant stage up to the adult size that you have now, it's because of cell division. For your wounds to heal, all because of cell division. Okay, so it's a very critical process that has to occur normally within the body. During formation of a new life, of course, after fertilization, the zygote has to go through multiple phases of division so that we can form an embryo. So cell division occurs at every level.
We're not getting it. That's what I'm not getting it. Audio.
Okay, Okelo Moses, you have an issue? Okay, I think you had mistakenly unmuted. Okay, so I was saying in your testicles, there are cells that are continuously dividing to form sperm cells. Okay, within the ovaries, there are cells that are continuously dividing to form mature egg cells.
Any audio from your side?
Okay, so someone is not getting what I'm saying. So let me pass on this communication again. If you happen not to see what is on the screen, if you happen not to hear what I'm saying, there's a simple solution. You just refresh your page, that page, just refresh it. You'll be able to see what is on the screen and you'll be able to hear me well. Okay? That's what you can do. If it continues to be like that, you just try switching positions, because different positions can have different network strength. Okay? If still it fails, there are some gadgets sometimes that can maybe lead to loss of networks. Probably. Okay? I don't know if it's true, but probably. Okay, let's continue. So cell cycle basically means the events that occur within your cells before cell division happens. That is cell cycle. And as I said, it's important for the cells to divide so that old and damaged cells can be replaced. This is very important. So just a few brief notes that you have to remember here. We all know that the nucleus contains the genetic material, and last time I told you that this genetic material can exist in form of thread-like structures that we call the chromatin. This chromatin can condense to form chromosomes. Chromosomes that can easily be separated into different daughter cells during cell division. Okay, so when a cell is about to divide, the chromatin condenses to form chromosomes. Okay, and every cell of your body has a total of 46 chromosomes. 46 chromosomes. If you talk about pairs, the pairs are 23. If you count from 1 to 23, you can see there are 23 pairs of chromosomes in every cell of your body. The 23rd pair is known as the sex chromosome pair because it determines your sex, determines your gender. If you're a man, you have XY. A lady has XX. Of course, these days, someone can be having XX, yet physically presenting himself as a lady. Someone can have XY, yet physically presenting herself as a gentleman. The world is becoming crazy these days. So half of these chromosomes are obtained from your mother, half from your father. Okay? You have to remember that. During formation of a new life, you all know the sperm cell combines with the egg cell to form the zygote. That zygote has chromosomes from the mother and chromosomes from the father. That zygote continues to divide so that every cell in your body will have equal number of chromosomes from the mother and equal number from the father. So you're seeing 46, for example. This is chromosome pair number 1. One chromosome is from the mother, the other is from the father. The same goes to all these. So I've said this 23rd pair is the sex chromosome pair, and these other ones, they are called autosomes. They contain genes that code for different characteristics from the body, whether skin color, those different traits on the body. They are going to be coded for by genes on these chromosomes. Okay? Chromosomes that we are also calling autosomes. All right. Now, the cell cycle. The cell cycle, you can appreciate it here, though the image is faint. But I'll say that's events that happen before the cell divides. And you can appreciate that we have different stages or phases. You're seeing G1, we have S, we have G2, we have M. G1, S, and G2, they are all going to be collectively contributing to what we call interphase. What is interphase? Interphase, it's the preparatory stage, okay? A stage that occurs to prepare a cell for division, okay? It occurs both in cells that are going to undergo mitosis and in cells that are going to undergo meiosis, okay? And during this interface, as I've said, we have G1, we have S, and we have G2 phases or sub-stages. And we have to identify the events that occur in G1, S, and G2. And after that, we shall look at what occurs in the M phase. So very simple. G stands for growth. So this is the first growth phase. This is the second growth phase. S stands for synthesis. Okay, so it's quite very easy. As I've said, this is just basic information which some of you already know, and it's very easy to understand. So G1. So in G1, what happens? Remember, this cell is going to divide. So if a cell, let's say, had 10 mitochondria, it will replicate them so that they can become 20. This cell is going to divide, so there should be duplication of organelles, so that after division, the daughter cells can have equal number of organelles. So organelle duplication occurs in G1, but also protein synthesis. We need proteins. For example, enzymes. You all know that enzymes are proteins, so we need enzymes that are going to be produced during protein synthesis, enzymes that are going to later help in the S phase. Remember, S means synthesis. Synthesis of what? Synthesis of new DNA. Without enzymes like DNA, like DNA ligase, okay, DNA helicase, we can't have any new DNA being formed during this phase. So as I said, in the S phase, we are aiming at forming new DNA, okay? And this is very important so that this DNA can be equally shared between the daughter cells. So before the S phase, this is how a single chromosome will appear. The chromosome will appear like this with just one DNA molecule or one DNA strand within it, one double DNA strand within it. This is one DNA molecule within this single chromosome. So what happens during the S phase, with the help of the enzyme like DNA helicase, DNA heligase, that single DNA molecule or that double-stranded DNA molecule is going to be splitted into two with the help of those enzymes, so that we can have a second DNA molecule. Now here there are two DNA molecules, and now since there are two DNA molecules, the shape of the chromosome will change so that it can accommodate the two DNA molecule, the two DNA molecules that are now present within the chromosome. Initially, the chromosome had just one arm, but now after duplication of the DNA, the chromosome will have two arms. These arms are also called chromatids or sister chromatids. The two sister chromatids make up a single chromosome. A chromosome which now has two DNA molecules after the synthesis phase. So that's the whole aim of the S phase. We want to duplicate DNA so that after or during division, it is equally shared between the daughter cells. In G2 phase, of course, the cell will duplicate more organelles that are required. For example, centrioles. Centrioles are very important organelles that are required during mitosis. Okay, microtubules, they will be duplicated. They are required to produce spindle fibers during mitosis. Okay, so more organelles are duplicated. Those that are going to be required during mitosis.
I'm not hearing anything. I don't know whether it's me only.
Is it a common issue? Everyone?
No, sir.
No.
No,
Yeah.
sir.
We are getting you. Well.
All right, let's mute. So the person who had said that you can't hear, kindly do what we said. Refresh your gadget, your page. Refresh that page. If it fails, switch the position. Okay. But also to those individuals that are facing those challenges, the good thing is that I'm recording, and you can always access this recording on the platform. Okay. Komakech, you are sending your colleague to go sit on a tree. That's not a good advice, Komakech. Okay.
Excuse me, sir.
So that's what happens during the G2 phase. Okay. Those important molecules needed for cell division are going to be produced. As simple as that. So in the M phase, what happens in the M phase? In the M phase, basically mitosis is going to happen. That's what M stands for. Stands for mitosis. And I'm very sure this is not something new to all of us. You all know what mitosis is. Okay. And I'm going to explain the same thing that you were explained to some days or months or years ago. So in the M phase, we have two processes that are happening here. Number one, karyokinesis. This means nuclear division. The nucleus is going to divide so that the contents can be separated to go into different daughter cells. That is karyokinesis. This process occurs through these different stages: prophase, metaphase, anaphase, and telophase. That is karyokinesis. After the nuclear splitting during these processes, now the cytoplasm and the membrane will also split. We call that cytokinesis, so that we can form two independent daughter cells. Okay, so now mitosis can also be called somatic cell division, because somatic cells, the term somatic cells is used to mean all body cells except the reproductive cells. The reproductive cells in a man, those are sperm cells. The reproductive cells in a lady are the egg cells or the ova. So in those cells, mitosis doesn't happen. Okay, mitosis only occurs in somatic cells, other body cells, okay, of the body. So during mitosis, of course, we know that prior to the process, the chromosomes duplicated, and that's how they are going to be looking like. That duplication occurred during the S phase. Okay. So in the early prophase, what happens? The centrioles, these are the centrioles, are going to move apart towards the opposite ends of the cell, and they are going to start producing what we call spindle fibers. But also, the nuclear membrane is going to begin disintegrating. It has to disintegrate so that the contents inside can have freedom to move towards the ends of the cell, because after the division, these components will be apart in different daughter cells. So the membrane has to start disintegrating at this point. So you can see here the spindle fibers. Spindle fibers are starting to form. The membrane is dissolving. It is disappearing. Okay. The nucleolus, which was a spherical body in the nucleus, it's also disappearing. Okay. After prophase, of course, we are going to go into metaphase. What happens in metaphase? In metaphase, the chromosomes are going to be arranged along the center of the cell. Okay, at this central line that you can call the metaphase plate or the equator of the cell. And these chromosomes, remember, they are going to be made up of two sister chromatids that are joining at a central point that we call the centromere. So what's going to happen is that these spindle fibers are going to be anchored at the opposite ends of the centromere, so that during anaphase, those spindle fibers contract and pull these chromatids apart towards the opposite ends of the cell. So you can see that very well here. Spindle fibers attach on the centromere of a particular chromosome. Okay. During anaphase, what happens? You can see now splitting of the centromere to allow the chromatids to move towards the opposite end of the cell. That is during anaphase. And during telophase, these chromosomes, or you can better call them chromatids, but it's also okay to call them chromosomes. They are going to be organized and surrounded by a new nuclear membrane, and the spindle fibers are going to start disappearing, and the cell is going to develop a cleavage furrow, and so that cleavage is going to form within or along the cell membrane. That cleavage furrow is going to be extending deep so that the cell can split into two to form two independent daughter cells. So that's how mitosis occurs without any kind of complication. Everything is very easy. That's how mitosis occurs. And as I said, this mitosis is very important in your body. Number one, early during formation of a new life, mitosis occurs in the cells that make up the zygote, so that we can form more cells that will form an embryo. And also during embryogenesis, as the embryo grows to form the fetus, still mitosis occurs so that the body cells and tissues can become duplicated, okay, to form a fully grown individual. When you get any kind of injury on your body, let's say the skin, the remaining cells of the body divide by mitosis to replace the injured cells. Okay? So mitosis is very relevant within your body. Okay, so now, if you look at the cell cycle, we've described what happens in the G1 stage, O phase, in the S, G2, and in the M phase. But there is another silent phase that we've not talked about. It's called the G0 or the G0 phase. This can also be called a quiescent phase. In this phase, the cell is going to be completely at rest. It's going to be at rest without any kind of cell division going on. Okay, there will be some processes that will continue happening in the cells, but it will not be dividing at all. That is the G0 stage, or the quiescent stage. Now, your cells go into this stage, for example, when there are less nutrients that are required for cell division. Okay. or when there are some genetic commands instructing this cell to permanently go into the gene node stage. Okay, as you can see here, either it can be because of the external factors. For example, if there are not enough nutrients, you know that cell division is a process that requires many nutrients. ATP is required after breakdown of the nutrients. So if the nutrients are not available, of course, cell division will not go on. The cell will undergo, or it will go into the gene node stage, the quiescent stage, the resting stage. Okay. Then secondly, there are some cells after maturation, they totally go into this stage and they lack the ability to continue dividing. Examples of these cells include the nerve cells. The nerve cells, specifically the neurons, those neurons do not have the capacity to divide. If a given part of your brain is injured, the remaining nerve cells cannot divide to replace the cells that have been lost. Those cells lack that capacity, okay, of division. They don't have enough requirements that can push them to go back into the cell cycle and continue with division. Okay. You all know that nerve cells are interconnected. They form neuronal networks that are very important in very many processes of the body. So imagine a cell, a nerve cell, going into cell division, yet there are some networks around it. It will be quite interfering with the networks. So naturally, nerve cells are made in a way that they cannot undergo cell division after complete maturation. They cannot. Okay, that's why the brain is a very delicate organ of the body. In case of any kind of injury, division of the remaining cells to replace the injured cells is very, very impossible. Okay, so the G0 phase or the G naught phase, we said it's a non-dividing state, but there are some human cells that are found or that are going to be within that phase. Some can be there temporarily, some can be there permanently. I have mentioned the nerve cells. There are cells that can be in this state permanently. They don't have the capacity to divide. Additionally, the cells of your muscles. If someone gets a muscle injury, say some part of the muscle of the thigh is injured severely, that injured part will heal by scar tissue formation, but no new muscle cells can be produced. That part will heal through fibrosis, through scar tissue formation. Same goes to your heart. The cells that make up the myocardium of the heart, if they are injured, that injured part of the heart just heals by fibrosis, and over time, that reduces the functionality of the heart, okay? Because we know that the heart is a very strong organ that has to pump. So if a given part of the heart just has fibrous tissue, but no cardiac muscle tissue, you can guess that over time the functionality of the heart is going to reduce. So these cells are in this phase permanently. But the liver cells can be in that phase, but if there is a a need to divide, the liver cells can divide, as we are going to mention later. So based on the ability to regenerate or ability to divide, overall, your body cells can be categorized into three. We have what we call labile cells. These are continuously dividing cells. They are cells that all the time are undergoing, they are going through the cell cycle dividing. Okay? For example, the cells of your GIT. The cells of your GIT are all the time undergoing division, and that's normal. Remember, we always eat things that are let's say, could be maybe solid food particles. Always digestion is going on. So probably there is always a possibility of some epithelial cells lining the gut to be injured. But those cells all the time are continuously replaced because the gut lining, the cells making up the gut lining are labile cells and they are continuously dividing. And that is good. The only disadvantage here is that such cells on the body that are continuously dividing are more prone to becoming cancerous, as we are going to clarify in the next few slides. Okay. We have what we call stable cells. Stable cells are cells that can divide when there is an appropriate signal. And within your body, the organ that I can mention to be containing cells that are called stable cells, that organ is the liver. It's made up of cells that are fully differentiated and not dividing, but they can divide when there is need. For example, if part of the liver is, let's say, removed, let's say you're told to donate part of your liver to someone, the remaining part of the liver, the cells in the remaining part of the liver have the capability to divide and replace the resected portion of the liver or the lost part of the liver. Okay, so that makes the liver to be unique. Okay, then we have what we call permanent cells. These are cells that don't undergo cell division once they are fully differentiated. For example, I've mentioned the neurons, the cardiac muscle cells, the skeletal muscle cells. Those cells are permanently differentiated. They are permanently, they are fully differentiated and they cannot easily undergo division. And I said if any part of the body containing those cells is injured, that part will just heal by fibrosis. Okay, and over time that reduces the functionality of that specific part of the body. This slide also, it's all about the same. We have the continuously dividing cells, and we've given examples of the epithelial cells lining your gut, the epithelial cells lining the skin. All those are continuously dividing cells. We have the permanent cells, and the good example here is the hepatocytes. And then the non-dividing or the permanent cells, here we have the cardiac myocytes, the neurons, the skeletal myocytes, all those are non-dividing permanent cells. All right, so this process on the cell cycle, normally it has to be regulated. It has to be regulated so that the risks of forming cancer cells is reduced. You all know cancer it's an illness associated with abnormal cell division. So if there is just one abnormal cell that has been formed as a result of maybe challenges faced during the cell cycle, that cell can become cancerous and it can divide to form many, many other cancer cells. Okay, leading to an illness that we call cancer overall. So we have to see how this cell cycle process can be controlled so that you can understand this concept of cancer. And it can also help you understand the pharmacology associated with management of cancer. Okay. Cell cycle control is very easy to understand. We've already looked at the phases of the cell cycle. You have the G1, we have the S phase, we have the G2 phase, we have the M phase. All these are phases. And in between these phases, there are what we call checkpoints. These checkpoints are very important to prevent abnormalities of cell division, to prevent cancer and many other abnormalities. You can think of these checkpoints to be like the same checkpoints that you find on roads. For example, you're driving. If your license is expired, you're not allowed to remain on the road. If your car, let's say, is in a terrible condition, you're not allowed to be on the road. Same thing here. If the cell that is yet to divide is abnormal in any way, if the DNA is mutated, that cell can't be allowed to continue with the cell cycle and divide. It can't be allowed. There are those checkpoints that will prevent that cell to continue going into the cell cycle or to continue into the M phase to divide. That will be prevented. Okay, so those checkpoints, you can see them being named here. We have the G1 checkpoint. Now this will screen the cell just before the next phase, which is the S phase. We have the G2 checkpoint. This will screen the cell just before the next phase, which is the M phase. We have the M checkpoint. This will screen the cell just before the end of mitosis. Okay, so as you can further see here, these are the checkpoints. We can call this the JS checkpoint because it is almost between the transition between the G1 to S phase. And you can also call it the restriction point. If the cell has any problem, if the DNA is mutated, if there is any challenge within the cell, that cell will not continue to division. It will not. Okay, and we are going to see what prevents the cell from continuing. Okay, then we have the G2M checkpoint, the checkpoint between G2 phase and the M phase. Now this is going to check if the DNA that has been duplicated is normal. Because remember, during the S phase, there was DNA duplication, and DNA duplication is a very long process that involves enzymes. There are very many events involved. The DNA is going to be splintered, and the different nucleotides are going to be attached together to form the new DNA molecule. So there are many chances of making errors during DNA synthesis or during DNA duplication. So those errors can be screened here, and if there are such errors, they can be corrected before the cell goes into the mitosis phase. Then you have the spindle checkpoint. Now this is going to check the attachment of the spindle fibers. We have seen that spindle fibers attach on the centromeres of a particular chromosome. If a spindle fiber is not well attached on the centromere, it can result into abnormalities, chromosome abnormalities. You all know that a chromosome has to splint so that one arm can go on one side of the cell, then the other on the opposite side of the cell. So if the spindle fibers are not fully anchored on the centromere, this full chromosome can migrate entirely to one side of the cell, resulting into chromosome abnormalities. You've heard of conditions like Down syndrome. Those individuals can have extra copies of chromosomes. Okay, we have Turner's syndrome. Those are ladies with who are missing some chromosomes. We have Klinefelter syndrome. There are very many, and I want you to read about all those kind of chromosome abnormalities. Read about the chromosome abnormalities and the various mechanisms that lead to those chromosome abnormalities. Okay, and the different features in individuals having those specific chromosome abnormalities. Read about them. Okay, so we've seen the checkpoints. Okay. Sister, please can you control yourself? Sister, I'm muting you and you are unmuting yourself. Kindly keep order. Control your gadget very well. Okay. So someone here was asking what is GIT in full. Okay, someone responded. So sometimes I don't get to see questions in the chat box. So if someone posts a question and you know the answer, you can reply. That's why I said always after a session, if I have not replied to any question that probably was in the chat box, you can always just text me. Okay, Nicholas, your hand is up.
Thank you very much. I don't know. I had something to actually ask about before you proceed on, because it's very important. Actually, on the growth one, we looked at enzymes that do help in cell division, and I would like to know some of them. Thank you.
Enzymes that help in cell division?
Enzymes, yes. In the G1, during the duplication of cell organelles, we look at protein synthesis. We need some enzymes during the protein synthesis. And these enzymes, I would like to know the example of them. Thank you.
Okay, so the enzymes that I so far mentioned, those are enzymes that help during DNA duplication, during the S phase. So during DNA duplication, there is an enzyme that will help to unwind or to split the DNA so that new DNA can easily be formed. Okay, the original DNA has to be split so that the bases are exposed in order to allow other bases to be attached so that we form a new DNA molecule. So the enzyme that unwinds the DNA is called DNA helicase. DNA helicase. Okay. Then I mentioned about DNA ligase. Now that one is going to be ligating, joining new bases to the DNA molecule so that we can form a complete new DNA structure. That's DNA ligase. Okay, there are very many enzymes. I can't mention all of them. There is topoisomerase enzyme. Now that one helps to stabilize the DNA during the process of unwinding, to prevent it from becoming entangled during unwinding. There are many enzymes, but as I mentioned, they are all going to be very important. There. Nicholas, is that clear to you?
Yeah, yes, please. Thank you.
Okay. So genes that help in the process of cell division or during the cell cycle. So far, we've seen that at these checkpoints, cells are going to be prohibited from continuing through the cell cycle if at all there is any problem. So what prevents these cells from continuing? The answer is there are some special proteins. Okay, we all know proteins are formed during protein synthesis. And we all know protein synthesis starts from DNA. And we all know that DNA has got special segments that we call genes. Genes that are going to code for particular proteins in the body. In this case, the proteins that are going to help in controlling the cell cycle and cell division. So these genes are broadly categorized into two. We have what we call proto-oncogenes and then tumor suppressor genes. The proto-oncogenes, these are going to be genes that are going to be expressed, or they are going to be coding for particular proteins that will trigger cell division to go on. Okay. These proto-oncogenes, they will code for particular proteins that will trigger cell division to go on. Okay. Proteins that will allow the cell to go through the cell cycle. Okay. Then on the other hand, we have what we call tumor suppressor genes. The tumor suppressor genes, these are going to be genes that are going to code for proteins that will shut down the cell cycle. You can imagine them to be like the brake pedals of your car. Those are the tumor suppressor genes. And then the proto-oncogenes, you can imagine them to be like the gas pedal of the car. Once you step on it, the car will accelerate. Once you step on the brake pedals, the car will stop. Okay, so these code for proteins that shut down the cell cycle. These code for proteins that allow the cell to go through the cell cycle. So as I have mentioned, the proto-oncogenes, they are going to encode for proteins that are going to allow the cell to go through the cell cycle, leading to cell growth, cell survival, and differentiation. Okay, and these proto-oncogenes can become mutated, unfortunately, and once they become mutated, they are going to become oncogenes. For example, the gas pedal of the car. We've seen that you can imagine to be like the proto-oncogenes. Once you step on it, the car will move. Okay, if there is any trouble with the gas pedal, things can go wrong in the car. Okay, same thing here. The proto-oncogenes are going to become, they can easily become mutated to become oncogenes, and these oncogenes, they promote uncontrolled cell division, which can easily lead to cancers. So you can imagine, using this animation here, the oncogenes, they just allow the cell to continue dividing even though there is a problem. For example, in this animation, when you are at the traffic lights, the red light tells you stop. If you continue when there is a red light, you can be knocked by cars coming from different directions. Oncogenes, they just tell the cell to continue dividing, and the cell as the cell continues to divide, there's a very high chance of this cell becoming cancerous over time. Then tumor suppressor genes, as we are going to see, these genes code for proteins that stop the cell from dividing. More about proto-oncogenes. We've said they are going to code or encode for proteins. These proteins are called cyclin proteins. These cyclin proteins are going to be produced in your cells, and they are going to be controlling cell cycle. There are different cyclin proteins for different phases of the cell cycle, okay? And they are going to be allowing your cell to go through the cell cycle. But these proteins don't act in isolation. Actually, they can't even act in isolation. For them to act, there should be an enzyme called cyclin-dependent kinase enzyme. This enzyme, it normally binds to this protein, the cyclin protein, and once it's bound to the cyclin protein, this whole complex becomes activated and result into continuation of the cell cycle. How? For example, in this image, we can see probably a cell that is about to start the S phase. Okay, this cell, without the cyclin protein of the S phase, this cell won't go into the S phase. But this cyclin protein has to be there so that this cell can cross the G1S checkpoint. Remember, there was that checkpoint, the checkpoint between G1 and S. Okay, if this protein is not there, the G1S cyclin protein, the cell won't enter the S phase. So it has to be there together with its associated enzyme, the CDK enzyme, and this whole complex will help in phosphorylating, or simple words you can use, you can say, will help in activating particular proteins that are responsible for the S phase to go on. For example, during the S phase, we say that DNA replication has to occur. Okay, for replication to occur, we need enzymes. For example, helicase, ligase. Those enzymes are inactive not until this complex is in place. If this complex is there, it activates those enzymes so that they can start the process of DNA replication, allow the S phase to go on smoothly. Okay, so that's how your body will control that. So then there's the M cyclin. The M cyclin, this M cyclin, it's going to allow your cell to smoothly go through the M phase. Its presence together with the associated enzyme will result into phosphorylation of special target proteins that can allow your cell form the spindle fibers, proteins that can allow your cells to condense, proteins that can allow your your chromosomes to condense, then your nuclear membrane to break down. We've seen that during the mitosis, spindle fibers have to form. The chromosomes have to condense so that they can be visible and easily separate. The nuclear membrane has to disintegrate. All this doesn't occur magically. There are control mechanisms in place to allow all these individual steps to go on, from mitosis to occur smoothly. Okay, so disintegration of this nuclear membrane has to happen because of has to happen after activation of some special proteins responsible to break down the nuclear membrane. So that's how these processes can be controlled. Then in this slide, you can see more. Control zones, all the checkpoints that we've already mentioned. This is the G1S checkpoint. This is the G2 checkpoint. Okay, to check the cell before mitosis. Then there is this is the M checkpoint, or the spindle checkpoint. So in the G1 checkpoint, we've already said if the cell has any problem with the DNA, it won't continue into the S phase. If the growth factors are not in place, it will not divide. It will just go into the G0 phase and rest up to when nutrients are availed into the cell or within the cell. If there is less cytoplasm, the cell has to accumulate enough cytoplasm to allow equal sharing during division. So if all these factors are not met, the cell won't cross this restriction point. It will be pushed to the G0 phase to rest. Then this is the G2 checkpoint. This will check the integrity of DNA. Remember, we have formed new DNA. We have to check if this new DNA is normal. Okay, that's why there is this G2 checkpoint. In this G2 checkpoint, all the newly formed DNA has to be checked. If it's normal without any challenge, there's a cyclin protein, the M cyclin protein, or what you can also call the mitosis promoting factor. It will come in to allow your cell to go through mitosis. And then towards the end of mitosis, we have the spindle checkpoint. And at this spindle checkpoint, there is a protein. It's a special cyclin protein. This cyclin protein is called the anaphase promoting complex. It's going to check if these spindle fibers are well attached on the centromeres so that the chromosomes can smoothly and correctly separate during anaphase. If the spindle fibers are not well attached, probably this cell may not go through this final phase of mitosis. Okay, so all these phases have to be controlled. The tumor suppressor genes. These genes are genes, as I said, they are going to code for formation of proteins that are going to prevent the cell from going through division. Okay, and I said these act as break signals, and they will stop your cell from dividing if there is any challenge. Okay, for example, as I related these genes to your brakes in the car. If you see any challenge inside, in front of the way, you just stop to prevent any trouble. Okay, but unfortunately, again, these genes can become mutated. If these genes are mutated, if the brakes of your car become faulty, the car will go wild and cause an accident. And in your cells, what will happen? The division process will go wild, resulting into cancer. Okay. So there are many good examples of tumor suppressor genes. The main one is the P53 gene. P stands for phosphoprotein, and this is the guardian of the genome because it's going to be found it's going to be expressed in many cells of your body, preventing the development of cancer. Okay, and this P53, how it acts. If it detects any abnormality in the DNA, it will stimulate the repair of that DNA. It will activate various enzymes that will stimulate the repair of the damaged DNA or the mutated DNA. If the repair can't be fixed, it will force this cell to either remain in the G1 phase or to go and rest in this G0 phase. Okay, if the repair is severe, this protein will stimulate the process of apoptosis. Apoptosis is the automatic cell death. Okay, that will be induced by this protein if at all there is severe DNA damage, damage that can't be fixed. Okay. So that's why in most types of cancers, it's noted that this p53 protein is not going to be there because of the mutation of the gene responsible for coding for this protein. If it's not there, a lot of errors can be uncorrected, and that can lead to cancers. There are many more other tumor suppressor genes. We have the BRCA1 and 2 genes. These genes are going to also be doing the same role of repairing damaged DNA and maintaining the genomic stability. Okay, and mutations of these genes is associated with high risk of developing breast cancer and ovarian cancer. Okay, then we have the APC gene. This APC gene is going to be coded within the cells lining the colon, and if this gene is mutated, someone can develop colon cancer. We have the RB1 gene. This gene is going to also be active in many cells of the body. If it's mutated, it's going to contribute to the development of this common cancer of the eye called retinoblastoma. Okay, there are many more. I just summarized a few, and all these are very important in your body. Okay, if that particular gene responsible for coding for a p53 protein or any of these tumor suppressor genes, if that gene is mutated, okay, it will result into failure of production of that particular protein. And if that particular protein is not formed, obviously this complex is not going to become activated. And if it's not activated, what's going to happen? This cell is going to pause in G1 phase. So if at all there is any kind of DNA damage, actions are going to be put forward so that these genes can help out in preventing abnormalities. But the main one is p53. It's the guardian of the genome. It's the main tumor suppressor protein that is coded for, and it exists in all cells of the body. If there's any kind of errors in the DNA, it will to inhibit formation of the complexes, and the cell will not divide. Okay, as you can also see here, normally this tumor suppressor gene, it's going to be coding for protein, for a tumor suppressor protein that will control cell division. If the gene is mutated, cells will just divide uncontrollably, resulting into cancer. Causes of mutations, I talked about them, I think it was yesterday, and I won't repeat them. There are many causes of mutations. All these can result into mutations of the tumor suppressor genes, and once those tumor suppressor genes are mutated, the chance of cancers arising is going to increase. So now we've been talking about cancer from the start of the session, but the question is it the same as tumor or they are different? Before I go to that, there is a hand that yes, Simon Patrick.
Thank you very much. In your explanation, do you mean to say that the tumor suppressor genes are the ones that can encode for programmed cell death? I got
I
interrupted a
said,
little bit with my network. That's
no,
why.
I said that genes encode for proteins, and tumor suppressor genes are going to encode for tumor suppressor proteins. For example, p53 protein, and that protein can easily stimulate or induce apoptosis in case it detects a cell with severely damaged DNA. Okay, that's what I stated, Patrick. Is it
All
clear?
right. Thank you.
Okay. So let's wind up. Have a physical session with the DMR group. Audio. So let's wind up and be able to attend to them. Okay, tumors. Tumors, they're just abnormal masses of tissue or lump of tissue that arises as a result of uncontrolled or abnormal cell division. These tumors can be in any part of the body. I think you've ever seen in the videos, you can see a person with a bump on the skin, and that bump is not causing any disease. That's a tumor, specifically a benign one, benign tumor. Someone can have a lump of tissue, a bump in any internal organ of the body, leading to an illness that we call cancer. That kind of tumor is called a malignant tumor. So we categorize tumors into two based on that. There are some tumors made up of cells just in one location, in one area of the body. Okay, cells that are not even spreading. Okay, that kind of tumor is a benign tumor. These benign tumors can be in any part of the body. I've just used an example of the skin because you can easily see those. Someone can have a lump around the ear, a lump around the neck. You've seen such individuals. They have the lump, but for many years they have been having it without any kind of disease. That's a benign tumor. Okay. The malignant tumors, they're made up of cells, abnormal cells that form a lump in a particular part of the body, and these cells have the ability to spread to other parts of the body. How do they spread? They spread through the bloodstream, okay, but also through the lymphatic system. Remember, if this is, for example, this is an organ, this is organ A, you know that this organ is going to receive blood through an artery, and it's going to be drained. It's going to be drained by a vein, and it's going to be also drained by some special lymphatic vessels. Okay, so this is how cancer is going. This is how these tumor cells are going to spread. They are going to spread either through blood, specifically through the veins, or through the lymphatic system. Cancer can spread through arteries. If this body organ is cancerous, or if it has abnormal cells of the body, tumor cells, these tumor cells can spread through the arterial blood. They are going to spread through the venous blood, because these are the blood vessels that are carrying blood away from this organ. So spread, or what we call metastasis, is going to occur through venous blood or through the lymphatic vessels, because we know lymph is also being drained away. So if there is any tumor cell here, it can easily be taken through the lymphatic system to other parts of the body. That's why always, if a patient has cancer, we always check the lymph nodes in the particular part of the body to get to a conclusion to confirm if there is spread of cancer. All right, benign tumors. These are non-cancerous, as I've explained. I don't want to repeat this. I've explained this already. They don't cause any serious health problems. but as they enlarge, they may press just nearby organs, leading to some minor symptoms. Okay. These benign tumors, they can become malignant. They can transform into malignant tumors if at all someone's immune system is weak. If the immune system is compromised, or if it's suppressed, it's possible for the benign tumor to become a malignant tumor. There are many other factors that can make a benign tumor to become a malignant tumor. You can make some more research on that. Okay, so tumors can be categorized based on the different cells that make them up. If the cells are epithelial cells, we call those carcinomas. Okay, if the cells are from connective tissues, we call that sarcomas. That's why you've heard of osteosarcoma, that is cancer of the bone. Okay, if the cells are cells of the lymphatic system, we call those lymphomas. As you continue with pathology, you'll learn about the Hodgkin's lymphomas and the various types. Okay, those are the most common ones. Then, if the cancer is affecting cells of the bone marrow, or specifically blood-forming cells or blood cells, we shall call that leukemia. If it's affecting the glia cells of the brain, we call that glioma. If it's the melanocytes of the skin, we call those melanomas. There are many different types based on the cell that you're talking about. Yeah, I just summarized a few. Okay, so as I said, this condition can occur in any part of the body, but mostly cancers affect parts of the body lined by epithelial cells, because epithelial cells are continuously dividing cells. So because of the fact that they are continuously dividing cells, sometimes there may be a possibility of a cell dividing without that division being regulated. Okay, because the cell is continuously dividing. So that can predispose such epithelial cells to cancer. Okay, so as I said, this knowledge is not only helpful in making you appreciate what cancer is, but also it will be very important in making you appreciate pharmacology. In pharmacology, you'll learn about drugs that target different sites of the cell cycle to stop cancer cells from dividing. Okay. All right. So I think I can end here. This session was all about cell cycle and cell division. But of course, you know that cell division there are two types of cell division broadly. We have mitosis and meiosis. I've already talked about mitosis. Again, I'm asking you to read about meiosis. Okay, meiosis is further divided into two, and it occurs in only some specific cells of the body. So read about it, and also most importantly, don't forget to read about the chromosome abnormalities. That's very important. Throughout the session, I always give out some reading areas that you can always research on in your free time. Okay, so whenever let's say if you meet a question in a new exam coming from such areas that are not in the notes, but I've talked about them, don't complain or don't blame yourself. Always you have to be attentive, note down what I say, so that you can make research on whatever assignment I give you. Okay. Okay, so I think that's the end of the session. We shall continue with this course unit next week, same time on Monday, Tuesday, and again Wednesday. And I believe before by next next week or before next week ends, we shall be in position to have concluded whatever we need to learn and this course unit. Okay, it's a very basic course unit, that's why I don't want it to take a lot of time. Okay. Okay, so I think we can end here. I can see some hands. Let's use 5 minutes for questions. Let me choose someone who has not asked. Alex. Alex, appear. Alex, you have a question?
Yes, sir. You meeting the DMR right now?
Yes, I'm meeting the DMR group because I take them through cardiorespiratory and digestive systems, and I'm meeting them at Block A just a few minutes from now. And I think we are going to use the second floor. We shall see the room that can fit all the numbers. Yes, I'm meeting them now. You wanted to... Are you part of the group?
Yes, I'm part of the group. I just wanted to know the exact time.
Yes. Just after this session, I'm going to Block A. Within 5 minutes, I'll be there. Then the BMI students, always you are going to be handled by Mr. Okiriza John. Always we divide the DMR from the BMI because we can through that we can easily manage the numbers, but the content is always the same. Okay, we just assign different groups, different lecturers, so that the numbers can easily be managed. Okay, we can't handle the whole of you at once. So the BMI students, I believe Mr. Okiriza has already given you your schedules that you'll be using. If not, I can send his number to you. I can send his contact to the class representative. He joins the BMI, the BDU, and the BPT, or the bachelor students. So class rep of the bachelor's group, do you have Mr. Okiriza's contact? Do you have it?
Yes, I have it.
Okay, that's good. So you can call him so that you can get to know the schedules that he will be always using for the lectures. Dear Mara students, let's link up at Block A so that we can do cardio right now. Have a good day.
Thank you, sir.